From Microbe TV, this is Immune, episode number 7, recorded on April 17th, 2018. I'm Vincent Racaniello, and you are listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hello again. It is snowing in Ithaca.
That's crazy.
As always. Not too cold. It's about 1 Celsius. But snow. It's not laying on the ground, so we're good. But
we had a huge downpour yesterday. Did you have that?
We did too. It was rain yesterday, thank goodness. But yeah, I'm ready for winter to be over.
We are 7 Celsius and cloudy here. Also joining us from Worcester, Ohio, Steph Langle.
Hey now, good to be back. Another Immune podcast. And we have similar weather, cold, snow, spring doesn't want to seem to come. But I don't know. I think when we live in these states, we just kind of, take it as is. And the southern states, so they don't even understand what it's like up here.
I love winter. I love winter sports. But by the time April comes, I'm done. It's enough.
Do you still love skiing?
I still love skiing, yes.
Good.
Ice skating I might avoid for a little while.
That's right.
If you didn't follow this, I had an ice skating accident this winter, so still recovering.
That's right. Yeah, not skiing, ice skating. Yeah,
you can have accidents in the summer too.
You can, not usually on ice unless you're training to be in the Olympics or something.
You could be. What is the thing where you go on skis behind a boat? Water
skiing. Boy, or wakeboarding, I think so.
I'm getting Alzheimer's.
Oh, hey.
Early onset, and I wonder what's causing it. Maybe we'll find out today. I hurt myself as a teenager water skiing and had a bad back for years. It finally, took 10 years for it to go away, but you can get hurt in the summer. All right, we have a follow-up email from last time. Follow-up on Immune is a little different because it's a month ago. But,
right.
Until if we do it because it's a weekly thing. But anyway, we have one about the last episode. Chaim writes, I have a publication on which I was one of 7, count them 7 co-first authors, Wu et al., Cell 2015. I think last time we had a paper with several.
Yeah,
there was like 3, I think, or 4 maybe.
In fact, all of my 6 first author papers so far have at least one other equal contribution author. For the most part, I am fine with this, as it means I can do much more interesting science than I could do on my own, especially as a computational biologist. However, I have received at least one negative grant review based on the notion that this makes it impossible to evaluate my unique contribution. Go figure. One more way in which my generation will have to change the paradigm, I suppose. Thanks for another great podcast. Well, it wasn't a paradigm in my generation. When I was training, and up until recently, there were no co-first author businesses because we hardly had any. I mean, I looked at my 2 postdoc papers the other day. It was just me and David Baltimore. So
yeah,
science is different now.
It's very different, and I think there's a lot more people involved to solve the problems we have. But you're right. Even like looking at papers from a long time ago, they're a single author papers.
Yeah.
Yes.
I don't know if that would get by a reviewer nowadays.
The problem is that when you have 20 authors, okay, then you figure the first author made the contribution, and then what if there are 3 first co-authors, and you're looking for a job? That makes it kind of hard. I remember once I was doing a podcast with Michael Cates, I guess, who has since been fired from UW. But I said, you have 50 authors on there. How do people get jobs? And later, one of the people in his lab said, I'm glad you said that because we have problems with it. And the thing is, often in these huge labs, they just throw everyone on the paper.
Right.
When maybe that's not warranted. Maybe you should just have fewer, depending on what they did. But
maybe we can ask from our audience, but I'd be curious if it's field specific. So she or he says they're a computational biologist, and I wonder if that also has a role in how that works.
Well, I know in physics it's very common to have up to 100 or so authors, and so everything is done in groups. Right.
Yeah.
I don't know. I don't know how they determine what the contribution is and how that weighs your ability to get a job.
Right.
Well, anyway, if you have ideas, let us know. This is one of the problems we have in publishing, that it's not easy to solve, amongst others. But we will not solve it here today on Immune. But what we will solve is your knowledge of something new. And it's now Cindy's turn to teach us immunology. So what do you have today for us?
Well, in our comments that we've had before and emails we've gotten, people have mentioned complement. And complement is one of those things that a lot of immunologists kind of tend to ignore, myself included when I was in graduate school. And then when I had to teach it, I actually had to learn it. And now I think it's absolutely fascinating, and I realize how important it is. And so I was going to give you a little bit of an introduction to complement, what it is, why we have it, how it works, how it's regulated. And then the paper I picked is quite interesting because it talks about a normal and somewhat pathologic role that complement can play. So complement we normally think of as a host defense molecule against infectious agents. But in fact, it has a normal role in homeostasis. So I don't know what you guys know about complement, but there's
a lot of different proteins.
There are a lot. So there are at least 30 different proteins, and it's well known for being involved in host defense. And it does this because the main job of complement is to attach a protein called C3 to the surface of a bacterium. You can even put it on a virus or a parasite. And once that protein C3 is attached to, or we called opsonized to a microbe, that's a way to identify that microbe as something infectious. And so when we think about innate and adaptive immunity, which are much more commonly thought of when we think about immunology, the adaptive part we know is really, really specific. I think in the past we've talked about it as the detective that looks for a fingerprint, right? It's a very unique characteristic of one particular pathogen. And the opposite side of that is the innate immune system, where you're recognizing something that's shared between a number of microorganisms. For example, lipopolysaccharide is on all gram-negative bacteria. So if you see LPS, you know you have gram-negative bacteria. But we can't encode the ability to see everything that could possibly invade us. So another mechanism to do this is to put a tag on anything that comes in. And so that tag is complement. And the complement protein that is tagged to invaders is called C3. And so that's the key crux of the whole complement cascade. And this is a covalent interaction. So once it's on, it doesn't come back off unless it gets cleaved off. And what this does is allows anything that comes into the body to get immediately tagged. And so you don't need the ability to see a conserved structure or a unique feature. You can tag this, phagocytose it, by like a macrophage in the tissue, and begin the process of recognition, digestion, and the ability to activate the other components of the immune system. So it's something that's always around.
How does it know? You say that when something comes in, how does it know that it's coming?
So it doesn't. So this is interesting. So there are 3 ways to activate complement. And the first way is called the alternative pathway. And
you see why everyone hates it?
I know. Exactly. This is why everyone hates immunology, right? Because the very first one that's activated is not the one that's named anything normal.
Let me ask you something before you go ahead. C3,
yes.
Does it bind everything?
It can bind
my own proteins?
It can. So it's just a chemical attack. So it's just a.
What is C3? A little protein?
It's a protein that has covered up basically. It has nucleophilic attack on in any carbon bond or any protein bond. So it's just a chemical reaction. It's not really any specific binding to anything. It's just when it's activated, it opens up the protein and it's able to chemically attack through nucleophilic attack to any chemical bond and just attach covalently.
Okay.
Yeah.
And you said there are 3 ways to activate.
No, there are 3 ways to activate it. The first way is the alternative pathway. And there's a lot. I give a whole lecture on all 3 of these pathways. We could talk about this for at least an hour, but I'm not going to do that. I will break it down to very simple terms. The alternative pathway we think of it as just a spontaneous activation And it happens all the time. And if it happens to happen very close to a microbial surface, the covalent bond will attach. So there's a lot more to it than that, but that's the basic idea. So these proteins are produced at high levels all the time, and there's this spontaneous low-level activation of this C3 protein. And if it happens, it's near a pathogen surface, there's this covalent attachment.
And if it's not near a pathogen,
then it just gets inactivated. Just it turns over and it gets inactivated and cleared. So it sort of happens all the time. Now, one could ask, well, why can't this happen to your own host cells and membranes? And the answer is yes. And I'll talk in a couple of minutes about some regulatory mechanisms, which are absolutely critically important, because if those are defective, then you have lots of diseases. Because if the C3 attaches to a host protein and is not kicked back off or inactivated, then it targets the host protein, the host surface of the cell for damage. And so the C3 is this critical part, and there's this alternative pathway that's spontaneous, sort of at low levels all the time. And then there are two other ways to activate this. One is called the lectin pathway. And so a lectin is a protein that binds to a sugar molecule or a carbohydrate. And so what this does, it recognizes unique carbohydrates on the surface of like bacteria. So mannose, high mannose molecules will be on the surface of bacteria. It's not very common on a host. So we have specific proteins that will then bind to that and recruit members of the complement cascade to activate this C3 specifically at that location. So the alternative pathway was this spontaneous, sort of happens here and there. And the other pathways are targeting it specifically to the microbe surface. So the lectin, there's a protein that binds to the surface and then recruits these complement proteins and activates C3 right at the surface to attach it. Now, the third pathway is the classical pathway, which makes no sense whatsoever, other than the fact that it was the very first one to be discovered.
So that was my question. Was that the first one, and then the alternative, right, was the next
one? So
one,
actually, the classical pathway was discovered first, and then the alternative pathway, and then the lectin pathway. But as far as the order in which they are activated and recruited in the host defense, the alternative would be first, then the lectin, then the classical, typically. Because the classical requires usually an antibody. And so we know that you have to have activation of the innate immune system to activate the adaptive immune system to get B cells to make antibodies, and then the antibodies combine to the microbes, and that recruits the classical pathway of complement. So typically there's an antibody that's going to recruit the complement component to activate the C3.
And depending on the isotype of antibody, does that then determine which one, whether it be classical or lectin? Because I know like IgA may be more likely to induce the lectin pathway, but maybe IgG is more classical.
So as I understand it, IgG and IgM are the two main antibodies that will activate the complement cascade. They both happen through the classical pathway. So the lectin pathway really involves proteins called mannose-binding lectin.
Right.
But the classical pathway will use IgG and IgM.
So the classical pathway uses this protein, which is a complement protein called C1q, right?
That's right. So this is a cool protein that looks sort of like a flower.
It looks very cool. Yeah.
Yeah. So it has a stalk region, and then it has little globular heads. And the little globular heads are what bind to the antibody that's on the surface of the microbe.
It kind of looks like a claw, those things, where you go down and pick a stuffed animal. That's what it looks like.
Now, this can also recognize certain patterns, right, besides antibody?
It can. So it can recognize also C-reactive protein, which is an acute phase protein that gets secreted during inflammatory responses.
Okay. So alternative lectin and classical, and then and these initial things which you told us about leads to a cascade of reactions generating different intermediates, right?
That's right. So there's a lot of proteins involved here, and there's protein, this protein cleaves that protein, cleaves this protein, activates that one. But the whole main point of this, whether you're going through the alternative, the lectin, or the classical pathway, the whole goal is to attach covalently the C3 molecule to the surface of whatever it is you're trying to tag. Mm-hmm. That's the main goal. And the reason why is because once you put C3 on, there are three things that can happen. The first thing is that is a tag that is recognized specifically by a complement receptor for phagocytosis and destruction. So the phagocytes will express complement receptors that can then bind to that C3 molecule specifically. So we call this opsonization. When the C3 is bound to the surface of the bacterium, for example, that's opsonized bacteria. And then the phagocyte will detect that C3 and phagocytose that microbe and kill it. So that's one of the main reasons why you want to tag those bacteria, for example, with complement, is to phagocytose them. And that's one, to kill them, and two, to then begin the process of digesting and presenting the peptides to activate the adaptive immune system. So there's that whole component too. So that's the first piece, the first of the three things that happens with complement. The second thing that happens is recruitment of a whole bunch of other proteins, C5, 6, 7, 8, and 9, which form something called the Mac attack.
Big Mac?
The Big Mac attack. So this is a series of proteins that come together and form a pore. But you need the C3 on first, then that activates and recruits all these other proteins and they form pores. And so the more C3 molecules you have bound to the surface, the more of these pores form. And when you form these pores, it's going to lyse the bacteria by osmotic lysis. This is just going to open them up, all the water is going to flow in, and they're going to explode.
If it were an envelope virus, could the same happen?
Same thing could happen, yep. So that's the second main way. So that's the direct lysis of the microbe. The first way was what we call the indirect. So you tag it and then it's eaten by the phagocyte. The second way is direct lysis. The third thing that happens that's really interesting is all these different cleavage events create fragments. And so that you get the active fragment like C3, which we actually call C3b, that attaches to the surface of the microbe. But there's a little piece that gets clipped off to activate that, and it's called C3a. Now, C3a and a couple of other of these fragments are called anaphylotoxins. And when those are released, they're highly inflammatory. They can change the vasculature within the area to allow recruitment of additional cells to the region of the inflammation. They can cause a leakage of the vasculature, so you get more complement proteins, antibodies, and other proteins into the tissue. And there's also swelling and associated inflammatory reaction that occurs with that. So the goal of that really is to allow recruitment of additional effector molecules to help with the infection, controlling the infection. So there's three ways to activate complement and three outcomes. The three ways are the alternative, the lectin, and the classical. The classical is the one we're going to talk about more with the paper because that's the one where you recruit the antibody first and then the classical components are recruited. And then there's three outcomes: the opsonization and phagocytosis, the direct lysis with the Mac attack, and then the anaphylotoxins that induce the inflammation. So those are really, in a nutshell, the main components of the complement cascade. And the one thing that we want to mention is that the classical pathway involves this thing that Vincent mentioned, which is the C1q that looks like the claw or whatever you want to call it. And that has two enzymes attached to it. So you often see C1q, C1r, and C1s. And we're getting into jargon now, but the main idea is there's always a set of cleavage events that happen. And so the C1q brings along with it its enzymes that can then cleave the other proteins in order to get to C3 and activate C3. Mm-hmm. So you'll see it often as a complex. But the C1 is multiple versions of this, multiple copies of this C1 protein come together to form this stalk and flower region. And that's what really is important for binding to the antibody, which is on the surface of the microbe.
Do you know why it's called complement?
Yes. So without going into too much detail, people were studying the ability of serum from rabbits to lyse bacteria. And they were trying to figure out what the components were. And they found that there was a heat labile component that was critical because if they heat inactivated, they didn't lyse the cells. And so they started adding back things that would complement the activity. And so, they did protein purification and isolation and little fragments of the initial gamish of proteins, and they figured out which ones did that, and they were called complement, and they started with 1 and 2 and 3 and 4, et cetera, et cetera, et cetera. And so complement 1 was the first one that they found would complement the activity to lyse red blood cells.
All right. And this happens where in our bodies? In the blood, in tissues, or what?
So it can happen anywhere in the body. It will often happen in tissues because there will be high levels of complement proteins around all the time in the tissues. So the liver is always pumping out lots of these complement proteins. And I think I read somewhere C3, the liver makes milligram quantities of C3 every day.
Mm-hmm.
So it's a huge, important part of host defense.
Cool.
And this is why in our assays in the lab, we heat and activate our serum so that any heat labile proteins don't potentially aggregate the immunoglobulins, making the reading of the assay more difficult. So it's kind of a neat way to think about it.
That's exactly right. Yeah. And so in addition to all of these 30 proteins that are involved in this whole complement cascade, I mentioned it's really important to regulate this whole process, because if you're willy-nilly activating these C3 proteins and these C1 proteins all over your body, you're going to start lysing your own host cells.
And that's so,
yeah, that is a problem. But there are several diseases that are associated with this. And so hereditary angioedema is one of them, and it's a rare disease. But if you have a mutation in a protein that's really important for regulating C1, it's called C1-INH or C1 inhibitory protein. If that's mutated, then you get this hereditary angioedema. And it has to do with activating C1 too much and then getting the anaphylotoxins activated. And it also has a role in other systems like the kallikrein system, which is just too complicated to get into right now. But it all leads to this severe inflammatory disease. Another one that I find that's really interesting is paroxysmal nocturnal hemoglobinuria. And so these people wake up in the morning and there's like blood in their pee and they freak out. But the reason why is because their red blood cells lyse. And then your kidneys, filter that overnight. And then when you pee in the morning, it's red. But this is because lots of the proteins that are involved in regulating complement have something called a GPI anchor, or a glycosyl phosphatidylinositol anchor. And so in PNH, they lack this enzyme. And so they aren't attaching the complement regulatory proteins on. So their red blood cells are vulnerable to being attacked by complement. And so that's how that happens. Yeah. And then one other disease that's interesting in mentioning is a complement 8 deficiency. So I mentioned that Big Mac attack complex. One of those proteins is complement 8 or C8. And if you lack this, then you can't make that Mac attack complex. And this is particularly important for capsulated bacteria. So Neisseria meningitidis, for example, has a capsule on it. And the Mac attack is really important for those particular bacteria. And so other parts of our immune system can compensate for other bacteria, and we don't see a problem, but we see a particular problem for those bacteria if you lack C8, because that is one of the main ways in which we combat those particular bacteria.
You may not know, I'm curious in that PHN, if it's lysing the red blood cells, do those patients have a harder time carrying oxygen? Are they anemic?
They do. So they do get anemic. Yeah.
Right. Interesting.
Yeah. And the last thing I wanted to mention about complement before we go on to the paper is that there are, in fact, 2 therapies that target complement. One is called eculizumab. So if you are interested in knowing about the naming of things, in the CAR T immune that we talked about, I mentioned a website in which you can look up how these things are named. And so MAB is a monoclonal antibody. And so eculizumab targets C5, which is also a component of this Mac attack complex. And so people who have diseases like PNH could take that antibody that will target C5 to prevent the lysis. And then there's one other one that I found that I didn't know before I looked this all up for this podcast was Cinryze, which is a C1-INH. So it's the purified protein. And I mentioned that one is one that's important for regulating C1 or complement 1. So you could actually take this complement 1 regulating enzyme if you have overactive complement. So we're starting to understand a lot more about regulation of complement, activation of complement, and now what we're starting to learn, and I learned more in the last few years, I didn't realize that complement had a completely other role in biology, because I always thought about it as tagging these invaders, opsonizing, lysing. But in fact, complement plays a critical role in normal processes and even in pathology in the brain. And so it's known that C3 deficient mice, for example, have defective clearance of damaged processes on neurons in the brain. And so it's thought that somehow complement and inflammation and the macrophages of the brain, which are called microglia, play some role in controlling this whole process. And if you paid attention to the title of this, we're going to talk about Alzheimer's disease. And so Alzheimer's disease is one of those diseases where the neuronal connections or synapses are damaged and you lose the ability to keep your memory. And so these people lose their memory slowly over time. And this is due, we now understand more, to not just an inflammation aspect, but there may be a critical role for complement in this whole process, and that it's normally there to prune these synapses and kind of keep everything in order. So you know how if you don't take care of a tree, it gets crazy, right? So you need to keep it trimmed and in order. And so the complement's important for that whole process. And without it being regulated properly, you can either get overactive trimming, and so you get loss of synapses where you need them, or you may lack the ability to trim them and you have too many synapses, and too many synapses in the brain is associated with epilepsy. So too few, we get memory defects and cognitive decline, and too much, we can get overactive signaling in the brain and epilepsy. So there's this whole idea of regulating the brain, and then that's a frontier that I'm not that familiar with. So we're going to talk more about the complement than on the brain part, but they're all connected together, literally. And so thinking about how these play a role in regulating that whole process is really important.
And it makes sense that there would be a regulatory mechanism by way to trim or prune, as you said, because when we're growing and we're developing maybe prepubertal or during those really important stages, maybe you have a lot of connections being made. You need the pruning aspect. But as you said, if you have overactivation in your older years, that could potentially lead to neurodegenerative diseases. And I love this paper that we're going to talk about because, and I don't know, Cindy, if you had a similar experience, but when we learned about complement, it was very how do I want to say, dry and dinosaur-like in the way that
boring.
People presented it. Boring. Like, this is something that was discovered this year, and just memorize these 30 proteins and that's it. And it was so difficult, honestly, because it was boring. And so this paper really helps to put it back in perspective that we have a lot to learn still on some of these classical, immunologic classical things we've learned, 80 years ago.
Exactly. So the title of the paper we're going to talk about is Complement and Microglia Mediate Early Synapse Loss in Alzheimer Mouse Models. And this was published 2 years ago, May 6, 2016 in
How
Science.
many citations do you think that it has amassed since 2016?
I didn't look. Did you look?
I did. I could be being dyslexic. It's either 314 or 341.
Wow.
It's quite a bit.
Yeah, that's a lot for one paper.
Mm-hmm.
Yeah. So the first author, Vincent, you like to say names. Do you want to say names?
Sure. Well, let me hide my other paper here. The first author is Soyeon Hong. Now let's see. Do we have co-first authors? Yes, we have. We have asterisks. Wow. So we have 3 co-first authors.
Oh, actually, there's one first author and two second authors. 2 second authors.
These authors contributed equally. Yeah, that's right. 2 co-second authors. It's interesting.
Yeah.
And then next we'll have co-third and fourth, Anyway, the first author is Soyeon Hong and Victoria Beha Glasser and Bianca Fonoyim, and those are the two second authors. And then the last person on this paper is Beth Stevens. And I just want to point out Ben Bars. Does anyone know Ben Bars?
I don't.
I don't either.
Well,
Is that Stanford?
Was, because he's dead now.
Oh, was.
Oh,
Sorry.
He died. He was a big glial cell person. And
oh, yeah. You know what? Yes, I just looked him up. So from what I understand, that he was a big proponent in the transgender community. Am I mistaken?
he was a woman, born a woman and became a guy. That's right.
Yes. And he really
right.
was.
And so he died at 63, unfortunately, had pancreatic cancer.
Right. But he meant a lot to a lot of people. So.
Yeah, totally. Yeah. And, this is, he was kind of a forged path.
Trailblazer.
Thank you.
Oh, I see.
Ben Bars. And, unfortunately he died. But he was a big microglia guy. And yes, so I presume Beth Stevens is the PI on this. There you go. Well, probably longer than you wanted.
Institute and of MIT and Harvard were the two key places. So
yeah, that's right. Harvard,
Boston.
You got a company in San Francisco.
Right.
You got Stanford, and also a company in Ireland.
Prothena Biosciences.
Yeah, I would guess that Alzheimer research would involve companies.
Yes.
Yeah.
Because you get a drug, man, you are
golden.
Yeah.
Golden.
It's a lot of motivation there.
Why do we develop Alzheimer's?
Why do we develop it?
Yeah, like we're
going to talk about.
We're going to talk about it. No, I mean not when we live long. It's increasing, and because we're living longer, right?
It is, yes.
When we died at 35, no one developed Alzheimer's.
I think you could ask the mutation rates that happen as we get older. Our polymerases, they make mistakes. They produce less, more, maybe.
I don't know. Maybe it has to do with oxidative
mistakes.
stress as well, right?
Yeah.
Yes.
Yep.
Definitely.
But we are not meant to live to 80 and 90. I think that's the problem. We did not evolve to live that long. Our ancestors certainly did not. So I think it's a problem. Modern medicine essentially allows us to live longer. Science, folks, science makes us live longer.
Yes, and better.
And so now we have to figure out how to deal with these chassis that were not meant to drive so many miles.
That's right.
All right.
Yeah. So, by way of introduction about Alzheimer's disease, I had to look all of this up. So if you have questions, I may or may not be able to answer them. But Alois Alzheimer, I'm not sure how I would say that, first described Alzheimer's disease, named after him, in 1907. And so it's characterized by atrophy in the brain and something specific called amyloid plaques, which probably a lot of people have heard about. And so these are deposits of a protein called A beta or amyloid beta, and it becomes aggregated and just accumulates in large amounts and is one of the key characteristic features of Alzheimer's disease and was thought to be the cause of the subsequent pathology. But we'll talk about maybe that's not entirely true. It's also characterized by something called neurofibrillary tangles, which is another aggregation of protein called tau. And so when these proteins accumulate, they cause loss of neurons and these synapses between neurons. And over time, as you lose your neurons, it leads to cognitive decline and loss of memory. And so this is not a trivial thing. Yes, this is primarily an age-related thing, but the global financial burden in 2018 is estimated to be $1 trillion. So this is a big, big deal. And it is the leading cause of dementia, which is the cognitive decline or loss of memory. And for a long time, this was just considered a degenerative disease. So the neurons just broke down and the inflammation was not brought into the picture until about the 1980s. So there was some initial ideas that, there are some markers of chronic inflammation, some cytokines in the blood and things. Maybe it's an immunological disease. And just to step aside here for a minute, pretty much any disease that you can come up with is going to have an immunological component unless it's like a structural defect during development. It's pretty amazing. So if you think your disease is not related to the immune system, I'll bet you
again.
It probably is. But so there were these hints that maybe chronic inflammation was associated with this disease. And in the 1990s, there started to be a lot more evidence. And so there was evidence of this inflammatory component. And believe it or not, even though we know now that the disease is mediated, at least in part, by the inflammation in the immune system, there's still no trials with anti-inflammatories that I know of. At least in this 2018 review that I read, they said there's still no trials in trying non-steroidal anti-inflammatories or other anti-inflammatories for this disease. So that's interesting. But so move ahead a little bit, and there have been these genome-wide association studies that have identified loci that are associated with Alzheimer's disease. And it turns out a number of them are related to the immune system, particularly with microglial function and complement. And so it's thought that these two play a key role in Alzheimer's disease pathogenesis. And up to this point, I think this paper in 2016, maybe a few other scattered papers had said similar things as this paper that we're going to talk about said. But for the most part, it was thought that once the disease starts, then these microglia and this complement become involved. So once you get these large deposits of this amyloid beta protein or this tau protein, then complement is recruited in the microglial cells, get activated, and then they do the damage, and that destroys the neurons. But there's sort of this flip in thought that maybe it's not exactly happening in that order. And so that's what the paper is going to get to. So this amyloid beta protein, or A beta, is generated from a precursor protein called amyloid precursor protein, and it gets cleaved. So this is a common thing, right? So the complement system I talked about cleaving proteins, and so this is also cleaved. And it gets cleaved by these gamma secretases called presenilin, 1 or 2. And so there are mutations in familial Alzheimer's disease, although that's not the sporadic is more common. So if it's familial, it would be genetic. If it's sporadic, they don't really know why you got it. But at least in the familial forms, there are mutations that lead to specific cleavage products of amyloid beta or A beta that are more likely to aggregate. So if you have these pre-existing mutations, then when your A beta gets cleaved, it's more likely to aggregate. So then you get more likely to get Alzheimer's disease. But like I said, there's very few of these that are actually familial, so more sporadic. And it's thought that they're linked with this A beta protein that gets accumulated. And if it's not cleared, it can be inflammatory. But what's clearing it and how it's inflammatory and why some people get it and some people don't is still a wide open field.
And do you know, in healthy individuals, is amyloid precursor protein, or the amyloid protein, does it serve a purpose? Because the paper
I should have looked this up. Yeah, so I've talked to people a number of years ago, and the consensus was, 10, 15 years ago, that no, we don't know what it does. Maybe one of our listeners can fill me in, but I'm not sure we fully know exactly what the protein does.
No, it's not known. No.
Okay, because it seems to be, we've only discussed it really in these degenerative diseases, but understanding the function could help us figure out why it accumulates.
There's
some ideas, but nothing really has been proven.
Okay. Yeah. I think if you knock it out, it's not a problem, or it's lethal. So one of the other ways, and they can't figure out what it does.
Sure.
So, okay. Yeah. But so this A beta protein can accumulate, but the other player we're going to talk about are microglia. And so microglia are near and dear to my favorite cell, which is the macrophage. So if you remember, macrophages come from different places. We talked about them coming from the yolk sac or from the bone marrow. And the microglial cells are actually the first cells to be seeded from the yolk sac. And so the yolk sac churns out these cells that are macrophage-like, and they go to the brain about 10 days into development, in the mouse at least, not in the human, but in the mouse, and then they stay there for life. And so they're called resident microglia. And they're called resident because there are additional monocytic macrophage-like cells that can be recruited into the brain when the brain gets inflamed. But those are the ones that are normally there. And so why do we have those cells there? Well, you could have infections in the brain, so you need those surveying innate immune cells there. But they also play a really important role in this whole idea of the pruning and keeping the tree in order, and not too many, not too few. And so they do this thing. And so they're really active. They're highly dynamic. They put out these processes that are really long, up to 80 microns in length. And it's estimated that all the microglia in the brain survey the entire brain every few hours.
Oh my gosh.
So that's pretty impressive because, they're these tiny little cells, but they survey the entire brain every couple of hours.
What are they looking for?
What are they looking for? They're looking for proteins, aggregated proteins. They're looking for dead cells. They're looking for synapses that are damaged that need to be cleared.
But they're not looking for pathogens, are they?
They look for pathogens as well. Yes. Yep. So, so they play this important role in both in the normal homeostasis and also the infection-induced responses in the brain. And so, so they do both of these things. But one of the interesting things is if you get large debris, and this is true for a macrophage as well, if they can't engulf it, and believe me, macrophages and microglia are extremely good at this. So I say macrophages can eat something that's larger than themselves.
Wow.
They do this by mobilizing internal membrane stores like from the ER and from lysosomes. So they can eat things that are really big, but if they can't get their mouth or whatever you want to call it around what they're trying to eat, they get frustrated. I guess like eating a giant sub that you can't get your mouth around. But so they get frustrated, and instead of trying to engulf it, they start spitting out and releasing their degrading enzymes extracellularly. So this is called frustrated phagocytosis. And when they do this, all those things they're releasing are highly inflammatory. So you can get this release of inflammation things. So the idea here is if you get these accumulations of aggregated proteins that are too big for the microglia to eat, they will start spitting out all these degrading enzymes to try and clear them, but that's going to cause inflammation in the local environment and damage the neurons nearby. And so that's how it was thought that this whole process was working. Large aggregates of proteins, the microglia try to clear it, they can't, they spit out their inflammatory agents, and then that damages the neurons. That was what was thought to be the process.
Sounds too tidy to me.
It is. It is. So it's more complicated than that. Yeah. So
it always is, right?
Exactly. So complement is what is complicating this even more. And so complement has been shown to be associated with these protein conglomerations or these aggregated proteins. And so there are C1 and C3, the ones we've talked about, are associated with these plaques or these accumulations of proteins. And the question is, who comes first, chicken and egg, right? Is the complement protein recruiting the other cells, or are they activating those cells? Are they being activated by the cells they secrete? Which comes first? And so this paper tries to address that a little bit by saying that the amyloid beta protein first will activate the complement, and then that causes damage, which then creates the whole storm. And the idea here is that the complement activation and the microglia activation are actually preceding the huge aggregates of protein. So it's turning the idea on its head from what was found previously. And I'm going to say this up front that this is one side, because there are a lot of conflicting data on the roles of complement in Alzheimer's disease. You can find papers that say if you knock out complement proteins, it's worse, and others that say if you knock out complement proteins in the mouse, it's better. So you've got different models of Alzheimer's disease. You've got ones where it's worse, one where it's better. But for the purpose of the discussion today, we're just going to say that the point of the paper is that there's early recruitment and activation of complement proteins and activation of microglial cells before anything else happens. And that it's this process that damages the synapses. And then that is contributing to the loss.
Of course, in complicating this further is the fact that these are mice. And can be different from what's happening in people for sure, right?
Yes. So there are studies in human cadaver brains where they've looked at, is C1q in the brains more in the brains of Alzheimer's disease patients? And the answer is yes. So there are correlations with the human biology as well, although it's not exactly the same. We can't do the studies in human. We have to do them in mice.
Right.
Yeah. So the last thing I want to mention is that I learned there are 3 types of neurons. Pretty much, there's sensory neurons, which we know really well because it's if something's hot, you pull away, right? But you pull away because you have motor neurons. So sensory neurons send information from the outside to the brain, and then motor neurons are when the brain is telling the outside what to do. So if you touch something hot, you'll pull away because your sensory neurons sent the signal to the brain, and the brain processed it and sent the information back to say, pull the hand away. And so the third neuron is an interneuron that connects those two. And so a synapse really is how those neurons touch each other. So you have to get the sensory neuron to touch the interneuron and the interneuron to touch the motor neuron. And if those connections are not correct, then the signal will not be propagated. And so then you also have your memory neurons and, things that are in your brain that store stuff. So if the synapses go away, you lose the ability to communicate. So that's really important. And in Alzheimer's disease, there's specific parts of the brain that I'm not so familiar with, but there's the hippocampus. And but if you lose your connections in those particular parts of the brain, that's when you start to get cognitive decline. And one of the important things is loss of the synapse connections is actually the strongest correlation with Alzheimer's disease, more than visual plaques and tangles and neuron loss. It's these connections, tight connections between the two neurons. And you have a presynaptic and a postsynaptic connection. So one's on the one sending the signal and one's on the one receiving the signal. And so what this group did was start to look at whether these synapses were there or not using an animal model of an Alzheimer's disease in a mouse. And so the mouse is called the J20 mouse, and it has human amyloid precursor protein that has mutations that have been associated with familial Alzheimer's disease. And it's a transgene that's expressed with a PDGF receptor promoter. And so these overproduce this human amyloid precursor protein that gets cleaved in the right way and starts to accumulate. And so these mice accumulate this A beta protein between 3 and 36 weeks. They develop the plaques that are characteristic of Alzheimer's disease between 5 and 7 months. And the neurons seem to be fine at first, around 6 weeks when the A beta starts to accumulate. But after a couple of more months, those neurons start to go away. And that correlates with spatial memory and learning deficits. So they use these specific tests that they put these mice in these mazes and they ask them whether they can find their treat. And then if they can remember how to find their treat, that's good. And if they forget and it takes them longer, then they have cognitive decline.
Is this eventually fatal in these mice?
I don't know if that specifically kills them, but in humans, it's thought that it's a contributor to death, although I don't know if they die specifically of just cognitive decline, because it would happen if it were in your the part of your brain that controls breathing and things like that. But I think that doesn't typically happen. It typically happens in the frontal lobe, which is where your memory is and speech.
Okay.
So they have this mouse model, and they take brain sections from this mouse and they stain it with presynaptic and postsynaptic markers, and they use something called Superresolution structured illumination microscopy, or SIM, which I think is really cool. And without going into too much detail, you take a filter and you just kind of turn it and turn it. You keep taking images like in a wide field microscope, but it use computational biology to look at whether the speckles from the floors are happening in the same thing when you shift it. And it allows you basically to double the physics defined diffraction limit. So you get much higher resolution. And, there's compromises between how many shifts you do and how long it takes you to image and things. But it's better. You get higher quality images than you would get traditionally with wide field or epifluorescence or confocal. And so we can put the link in the show notes to if you're interested more in this type of microscopy. But the pictures in this paper are really good. I highly recommend you go see them.
Have either of you ever worked with this type of microscopy before?
No.
I haven't done sim microscopy. I've done a lot of confocal.
Sure.
But yeah, so basically what they show is that in this mouse model, they lose the synapses. So more so the postsynaptic synapse parts just go away. And so the question is why? And so they asked, is complement correlated with this? And what they see is the C1q molecule, which is important in the classical pathway, is highly accumulated at these regions. And it happens over time, which means this is not a congenital defect or a congenital problem. So at 3 weeks, the wild types and the J20 have the exact same staining patterns with C1q. But over time, C1q starts to accumulate and accumulate and accumulate in the critical regions of the brain and the Alzheimer's disease mouse. And cool thing is they use more than one model, because one could say this is an artificial model. What does this have to do with real disease? But they've used multiple models, and people have looked for C1q in human brains as well. And so they see this correlation with this postsynaptic marker with the C1q. And so their idea is that maybe C1q is being recruited to these synapses for their clearance. And so this is aberrantly happening. Normally, those synapses would be fine, but somehow they get marked for clearance by complement, and then they're going to be erased.
Right.
So they start to look at whether the A beta is involved in this whole process. And so they look for accumulation of A beta. Now, Steph, you looked this up a little bit. The A beta goes from a monomer to these plaques through various stages, right?
Yeah, it's interesting. I mean, they're really defined, these aberrant protein foldings. It happens in a lot of different types of diseases, even prion diseases, is when you have a protein you have a lot of it, and maybe it's misfolded, and they're in their own little, they're single individual monomers of this protein. But if you increase the number, maybe they then come in more contact and they start to form these oligomers, which are just bunches of those monomers. And then when you continue to increase the concentration to kind of get over that entropy of interactions of the proteins, then they form aggregates. And then those aggregates, based on what I was reading, they open up conformationally. And if you can nucleate, if you can find like a seeding event, it's really interesting, of one particular aggregate that can start to form these fibrils. And it's these fibrils that are really long, and those overlaid form the plaques. So it's interesting how a monomer will do nothing. However, an oligomer can cause problems, but then you get past that and the actual fibrils may not be toxic. It may be this intermediate products that are the toxic products that then just end up forming the fibrils. So, yeah.
Yeah. So what they're wondering is there increases in the A beta protein, and does that happen before the plaques, which is what you would predict based on that process that you just described. And so, and the question is, could the complement be increased, the protein expression and accumulation be increased in response to the monomeric or oligomeric, those early stages of A beta production? And so one of the ways in which they did this was they treated with something called compound E, which prevents the cleavage of the amyloid precursor protein, so you don't get generation of A beta. And when they do this in that J20 mouse, the C1q goes away. So somehow you need the cleavage of the amyloid precursor protein and the production of at least those monomeric early A beta proteins in order to get recruitment of the complement. So there seems to be this progression that needs to happen. And interestingly, the C1q protein was associated with microglia. So it seems that the microglia in the brain can produce the complement protein, and they do that after you start to accumulate this A beta protein.
So we found out the chicken and egg question then.
Correct.
AB protein, then C1q.
Yeah, and they nailed that down even more, because then they injected the A beta protein and cause the synapse loss and accumulation of C1q.
Why do they have to inject because the mice are producing it?
So
right?
they actually did that in the wild.
Oh, okay, got it.
So they walked away from the Alzheimer's disease mouse for a minute and just said, in a wild type mouse, what if we put this A beta, oligomeric A beta, so the second stage, into the mouse? Does that induce it just on its own? And it does.
How do they inject that into the mice?
So they did this stereotactic injection into the ventricle of the brain.
Okay.
Yeah.
Cool.
Yeah. So that connects it. So they connected this a little bit, right? So if you have production of A beta, you have recruitment of C1q. If you prevent the production of A beta, you prevent the accumulation of C1q. And if you inject A beta, you can induce C1q. So it all connects together. But the question then is it absolutely necessary for the downstream events? So then what they did was they inject A beta into a wild type mouse, or a wild type or a mouse that lacks C1q. And when they do that, the A beta no longer induces the synapse loss in the absence of C1q. So that connects it back together. And they did it one more way, where they used an acute antibody blocking of C1q. There's an antibody that targets C1q. When they inject that in, they prevent the synapse loss induced by A beta. So they did this a whole bunch of different ways, and they show very clearly that there's this connection. There's an early production of A beta that induces the production and accumulation of C1q, and then that's required for the synapse loss.
So is that a way to treat human Alzheimer's with antibody to C1q? Does this have to be injected into the brain? Did they put that?
Yes. So one, yes, it has to be injected into the brain. That's a problem. But two, remember, this is really early.
Yeah. Because
this is a model where they spontaneously develop this disease months later, and they start treating them very early. So
maybe too late. It may be by the time you see, if you already see symptoms in humans, it's too late.
It's very late once you start to see symptoms. That's right.
All right.
So coming back to our complement lesson, we learned that C1q is the classical pathway, and that no matter which pathway you try to activate, the goal is to attach C3, right? So the next thing they just ask is, do you need C3 for this whole process? Because the presumption is you do, but maybe C1q somehow is binding to another protein, or binds to a receptor for complement 1, or it's working some other way. Is C3 involved in this whole process? So we already know C3 is important during development for that whole pruning the tree of the neurons. But what they found was, if they injected the A beta in the wild type mice, they could cause accumulation of C3, and it didn't happen in C1q knockout mice or mice treated with antibodies to C1q. So the idea here is that A beta comes first, activates C1q, C1q activates C3, the typical classical pathway. And then the next question is, well, what does all that do? And we talked about, when you activate complement, there's 3 things that happen. You get the MAC attack, you get the anaphylotoxis, and you get phagocytosis. And so they asked then, is this tagging of these postsynaptic synapses really important, and do the microglia come in and eat them? Are they actually clearing those synapses, and that's what's causing the problem? And they do this in a really, really cool way. So they use a transgenic mouse that expresses a marker fused to GFP that's found on the postsynaptic synapse. And if the microglia have eaten those, they turn green. And so they can stain the microglia, and they can see if they've eaten these little green synapses. And they find that in the J20 mice, they do. They eat those synapses. And they also see that those synapses that get eaten, that GFP signal goes to the lysosome. So it's for degradation. So it's pruning those synapses, eating them, and pulling them away. And this is all dependent on the complement.
Yeah, it's a cool experiment. I like that a lot.
Yeah, I thought that was really. And then really the last thing they do is they asked if they could just inject A beta in the tail vein and look for synapse loss in the brain. I thought this was less of an interesting experiment because really all the A beta is being generated in the brain, as far as I understand. And so you have to cross the blood-brain barrier and all of this. But maybe it was an experiment that a reviewer asked for. But I think in general, they've pretty clearly shown that you get this early generation of the A beta, and it's either monomeric or oligomeric that causes the problem. And once that's made, that recruits the C1 through the classical pathway. And what we left out there is that antibody dependent or not? I don't know. I mean, this might be a unique way to activate C1q. But
that's a good point, right? Is it really classical if there's no antibodies involved?
Right. Yeah. Huh. So anyway, so C1q gets there. It recruits C3. The C3 tagging allows the microglia to recognize the synapse and clear it. And this is important for early loss of the synapses before the classical characteristic definitive markers of Alzheimer's disease happen, where there's these large accumulations of the plaques and the tangles.
You said there are other views, right? This is not the only one, but if we just took this as a key in the pathogenesis of Alzheimer's, what does it suggest? Any interventions at this point, or is it too early?
Well, I would assume if you could block the activation of C1 in the brain, you could block the early activation.
But you have to you have to know this early on. That's the problem. Right?
That's the problem.
We'd have to write some biomarker that's upregulated, because I think the real big question too is what starts this cascade of the APP protein and then the amyloid protein forming these monomers and oligomers, like whatever starts that process is probably what we need to identify to be able to get it early, because otherwise. I mean, there's ways I was reading in the back of the retina. They are now looking at plaques in the back of the retina as a predictor for plaques in the brain. But again, those plaques would be the end. That would be too late.
Mm-hmm. Yeah. Once you eat the synapse, you can't get it back, right?
You can't. Nope.
So you do have to go very early. You have to, as Steph said, you have to find out why these oligomers are forming to begin with.
Right.
But I mean, you have to have some markers early on, and you have to understand the basic biology better, because right now we can't see this until it's symptomatic, and that's already too late, right?
That's right. Because what this paper is saying is that once you have plaques, which are visible, I think, on MRI or PET scanning, that's too late, because this complement's already been activated, has already started to eat away the synapses.
So essentially, you have to figure out what is causing the oligomerization and the fibro formation, et cetera.
That's right.
And then at the age where this is typically coming on, you have a small molecule that's been developed. I don't know what it's going to target, that you would then start taking prophylactically.
Yeah,
I would think that's the way to deal with this.
So I think that a lot of the oligomerization, and especially the plaque development, has a component of ER stress in there as well.
Oh, interesting.
So
So oxidative stress may be an issue.
I think so too. Yeah.
Yeah, because we have a lot of that. I have to tell you guys. I'm reading this book, Oxygen, by Nick Lane, which is just amazing. And he talks a lot about oxidative stress. And I got to tell you these numbers here. Look at this. 1 to 2% of the total oxygen that is consumed by our cells escapes as superoxide radicals.
Wow. Really?
Which is amazing. And we breathe in 1/4 of a liter of oxygen each minute. So we produce 1.7 kilograms of superoxide each year, which is in turn converted to hydrogen peroxide and hydroxyl radicals, which are really nasty. They will just, rip electrons from whatever's next to them.
Oh, yeah.
In a full day, every cell in our body generates 4 million hydroxyl radicals.
Wow.
Many of these are neutralized, and of course, if they happen to have caused DNA damage, that can be fixed. But
I'm going to go drink my red wine.
but not everything is fixed, right? And that's why, and they say in this book, DNA damage caused by these reactive species result in several thousand DNA hits per cell per day. Per cell, though.
Wow.
And of course, these radicals, peroxides and hydroxyls, also hit proteins, right?
Yeah,
so
yes.
this is happening in our brain, and that's possibly part of this whole thing, right?
Could be.
So you have to early in life deal with these radicals, and having a lot of fruit is part of it. It sounds wacky, but fruit is a good source of antioxidants.
But man, has this generated a whole industry of pseudoscience when you think about, antioxidants as supplements, or when you Google oxygen radicals, a lot of nonsense pops up.
Well, that's what Linus Pauling did in the end of his life. He was eating grams of vitamin C a day, but it's not just vitamin C. It's other stuff.
Yeah. It's probably just peeing that out.
Oh yeah, they're over so many milligrams, it just pees out. Absolutely. Right. Totally. So it's just not, it's whatever else is in many different kinds of fruits and vegetables, really. But it's still a soft science, and we need to know exactly what's going on. But man, it's very interesting.
This is a great paper. I'm glad you chose this because it got me excited about complement again, which I can't really say. Can't really say I was really too excited about.
Yeah, definitely. It's cool. I mean, this is very clear. The results are very clear. And, you have to... you have, unfortunately, the only way you can study people is by cadavers, right, after they've died of this. But it would be interesting to go in there and try and correlate some of these observations in the brains, right?
Particularly in early onset dementia. I'm thinking about that UConn basketball coach whose name I'm now forgetting. I mean, she succumbed to dementia when I think she was in her 50s, maybe, early 60s. So maybe in those people, maybe there are biomarkers that we could identify.
Mm-hmm.
Great job, Cindy. I mean, I think it was really well done with the intro, and it led right into it. So I think people will like it.
I forgot the book. Do you guys know the name of the book about Alice? There was a movie where it was a scientist who lost her.
Yes. Yeah, because there was a movie.
Just Alice. Ask Alice. Alice.
The actress that was in that movie has red hair. I'll look it up. I know exactly what you're talking about. I think it may have won something. But yeah, she was a scientist and then got Alzheimer's. Did she study it? I can't remember.
Yeah, it's based on a real story. Still Alice.
Yep.
Yeah. Julianne Moore.
There you go, Julianne Moore. Yep.
Yeah. The book is stellar. If you haven't read it, it really follows her, and the way it's written, she talks as she's losing her mind. It's really cool. Wow. Anyway, I know we have to finish up, but yeah.
Well, thanks.
Do you want to read emails?
That was great. Let's do some picks. That's what we have time for. Yeah. Steph, what do you have?
Okay, so two things quickly. There was a recent Science Magazine article about the use of social media for social change in science, and that people now have a platform they didn't have before. And there's a lot of conversations back and forth whether people should be involved on social media. And I think it addresses some of the current conversations going on back and forth about that. And I think it is true that using social media, and I'm sure as you all do, or just communication to reach people, there's good and bad ways to do it. And of course, not everyone has to, but it's definitely a way to enact social change. So there's that. And then the second one, when I was reading for this episode, there's a really good video by Dr. David Agard at UCSF. And he is talking about the SIM, the microscopy, structured illumination.
And we'll do a much better job than what I said.
It's a good video. So yeah, take a look.
What's one of those iBiology videos?
Yeah. Yep.
Cool.
Very good. Nice. Cindy, what do you have?
Yeah, so I was thinking as I put this in that I don't know if I mentioned this already in a previous one. I should have gone back and looked, but I put on this 314 Action, because 314 Action is a PAC that supports scientists who want to run for political office, and it reminded me because I was in Albany this weekend at the New York World Democratic Convention, and there was a lot of talk about issues like sexual assault, women's rights, LGBTQ rights, budget, but almost nothing about science. And I was like, what are people doing? Yeah. So we really need the word out there. We need people talking about this. And 314 is cool because, as you know, it's the first 3 digits of pi. And so it's this PAC, and they're supporting all these people who are willing to run for office. And I'm not saying that our listeners should necessarily run for office, but it gives you ideas of candidates that might be running in your area, or people you might want to support, or at least put the the word out on social media to help them in their campaigns because it matters. And they have something on their website that says, a quote from Albert Einstein that says, "I do not share your view that the scientists should observe silence in political matters." And that's something that means a lot to me because I don't think we can be quiet. I think we need to be the squeaky wheels. I think we need people to run. I think we need to make science a priority.
Yeah, I get a daily email from 314. They're always outraged, which they should be, right?
Yeah.
It's the crazy stuff that's happening nowadays. It's just ridiculous. So I gave them money because it's the way I can help, right?
Right.
I think it helps. I can't go knock on doors, but giving
you don't want to run to be a congressperson?
No, that's not my strength. I'm too volatile and too opinionated. I can't keep my opinions back.
Then you'd be perfect for it.
No, I can't. You have to massage everyone left and right and not offend
Right,
anyone.
dude.
Although,
Well,
wait, we have a president who did that. What the heck?
Yeah,
But he may not have gotten in legally. Who knows?
Yeah, I don't, we may be entering a different era. Who knows?
No, please don't say that. It's horrible. You're right.
No, I'm sorry.
But it's true. We think maybe our democracy is going to end soon. Who knows?
No. I think it has strength and persistence, but we have to be vigilant, as Cindy said.
We do. But money can make a big difference, Anyway, I have a pick, which is a nice article at PLOS Biology, which is open access, so you can read it. It's called "Making America Great Again Requires Acting on Scientific Knowledge." It's by Elizabeth Hadley, and it's a story of how she grew up reading an article. When I was 15 years old, she writes, I learned that chlorofluorocarbons could be a problem, and then she traces her whole growing up and entering science and tells stories of how throughout her life, getting scientific facts was really important. And at the end, the key is the irony is hard to miss. The mantra these days is to make America great again. But a key feature of those bygone days was listening to the lessons that science taught and acting upon them to make a better planet. Today, it is the trepidation that even though we know we might not act, and that makes me very worried. It's a nice little article, which I love, twisting around the great nonsense thing and saying, make science great again.
But notice that she tells stories.
Tells stories, always.
Yeah.
And it's her personal story. Yeah.
But that's the key. We need to tell more stories. As scientists, we tend to be stuck on the facts and not put that into context, like learning complement, in a very dry fashion. But you put a context to it, it makes more sense. It ignites people and excites people. That's why we do this podcast. That's why we talk about this. It's why we tell these stories. We need to ignite people about science.
And that is Immune number 7. You can find it at microbe.tv/immune, and you can find it on any of your podcast apps on your phone or tablet. Just subscribe and send us your questions and comments, immune@microbe.tv. And if you'd like to help us financially, microbe.tv/contribute. Cindy Leifer is at Cornell University.
Thank you.
Yeah, you're welcome. You can find Cindy on Twitter, Cindy Leifer. Steph Langle is at Ohio State University. Thanks, Steph.
Thank you. I loved it. It's great.
Stephanie Langle on Twitter. I'm Vincent Racaniello. You can find me at virology.ws or Prof VRR on Twitter. Music on Immune is by Steve Neal, stevenealpercussion.com. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.